In the light of the rapid climate changes and increasing the world population, so NatGenCrop aims at improving stress tolerance in two major crop species, specifically tomato and pepper, and designing a new strategy for maintaining high yield and food quality under a harsh environment like salinity and drought.
The project has several scientific research packages. The first one is related to identification of genes that modulate important traits such as fruit, size, shape and color in tomato and pepper. The second one is related to identification of genes that modulate abiotic stress tolerance. And the third scientific work package is related to biotic stress tolerance, response to pathogens, etc.
One of the advantages of the project is that it gives us the chance actually to implement various research techniques and to employ researchers with different expertise within the new team.
2023 was very dynamic for our project because we formed our team, scientific team.
We are very proud that we were able to recruit a prominent scientist from the Max Planck Institute of Molecular Plant Physiology, Dr. Saleh Al-Sheikh, who is coming to head this department, Crop Quantitative Genetics.
And of course, this is also not easy part, I think, for any project or, recruiting people as well, selecting people really to work on the project. And this is something we have really been doing in the last 2 years.
And altogether, we established a very good team who is already producing good results. For example, for the last year we published 2 high-profile papers and many more are expected in the next years.
My responsibility in NatGenCrop project is to take care of plants for research. We cultivate peppers and tomatoes in our project, and I am responsible for whole process of cultivation, which means it's production of seedlings, protection of the plants, fertilization and implementation of drought stress.
Last year we grew a lot of crops and we met the targets of producing a GWAS and a biopanel of tomatoes and peppers. We analyzed a lot of phenotypic data and we recognized candidate genes that are related to the resistance of the plants to drought stress.
So plant material used in the project is 2 populations from GWAS panel, which is 180 lines of peppers and 152 lines of tomatoes.
With really a large diversity in fruit size, shape and color.
Also, the research is going on 2 Bill's populations, backcrossed inbreed lines, which are 56 lines of tomatoes and 110 lines of peppers.
And our purpose is to phenotype and genotype them in a way that we are trying to determine which genotypic characteristics actually, which mutations in these lines are responsible for what phenotypic characteristic of the plant.
We analyzed the plant growth, the plant height, the leaves. We also analyzed their fruits and their characteristics. And we came to recognize a number of genes that may be potentially responsible for helping the plants endure drought stress.
Bulgaria is known to be one of the hotspots worldwide about the genetic diversity of tomatoes. So we all know that this famous Bulgarian pink tomato is believed to originate here, and it's very tasty and it's used widely. So our purpose is to find such interesting varieties of phenotypes of these different characteristics of the plants and to introduce them in our modern sorts, which are very famous and very economically important.
One of the important also aspects we are really looking at using tomato and the pepper is the diseases resistance. You can see some of the genotypes, they are really susceptible to diseases, they are affected by diseases, while others they are really resistant, and this is one of the traits as well we are really looking at for the gene or the genes they are really regulating such kind of important traits. Basically, after really collecting the fruits from the greenhouse or the field, we bring them back to the lab. First, we take pictures and also the weight. also the external color. Also using the tools and the high throughput after taking the pictures, we can quantify the size and the shape and use a more automatic way to look at this trait quite fast comparing just do it manually, especially when you have a large number of samples. And then we cut them to small pieces for further analysis. Basically the metabolite profiling, which is really a very important part of the project, is looking at the quality. And the second part as well, using such kind of materials to do some omics analysis, like transcriptomics in combination with the metabolite profiling. This is for transcriptomics and metabolomics. We should have a liquid nitrogen and do snap frozen.
As part of my role in the NatGenCrop project, I will be processing the various samples from tomato and pepper that are provided by the research team. And I will be analyzing them using two methods. One of them is GCMS, which is gas chromatography mass spectrometry, and the other is LCMS, which is liquid chromatography mass spectrometry.
The ICP-MS uses inductively coupled plasma in order to atomize and ionize the sample. This gives us the opportunity to detect really small amounts of either metals or non-metals in the range of like part per trillion. And this is the way that we are going to implement it in our project, NatGenCrop, because you know that people are usually using different type of supplements, like if they have some type of deficiency. So instead of taking food supplements, we can supplement our diets with vegetable crops which are rich in different micro and micronutrients.
As a scientific part for this project, it's not only looking at really selecting the genotypes, but as we are really scientists, we would like really to find novel genes and novel regulators from doing this QTL mapping and analysis. And so the transformations facilities, the cloning facilities in the lab, also they are really available and really already set up to start and working on in this project. And one interesting thing and important is really, this is really relatively a new institute, so you have really the very basic large space, so you can really grow large number of experiments. This is really impossible to do it with other places, so this is really very, very important.
This year we are repeating our experiment just to have more thorough data about the candidate lines for later research, which are more resistant to drought.
And ultimately our goal is to find application for this knowledge in agriculture by producing plant and pepper varieties which have the drought resistance phenotype.
In conclusion, I can say that this project gives us the resources to improve the research capacity of our center so that we try to solve a real-world problem that is important not only for researchers but also for farmers and the society as a whole.
So this is related to food security and sustainable agriculture.
Our project is also very important because it will provide us the opportunity to educate several new PhD students in various disciplines so that they become basically the new generation of molecular biologists in Plovdiv.